Slaughterhouse blood is an unavoidable byproduct of the meat processing industry. Blood is a major source of protein, but it is also a source of bioactive peptides which can be used in different applications to modulate various physiological and metabolic processes. 30% of slaughterhouse blood is further utilized within the food industry as an additive in foods such as blood sausage and black pudding. Last year in Canada, over 57 million kilograms of bovine blood was disposed of without further processing. The high solid content of blood and the high chemical oxygen demand increase its negative impact on the environment and represent an incredible waste of potential bioactive compounds. Given these characteristics, the generation of bioactive peptides from slaughterhouse blood is justified and needed. The bioactive peptides found in cow, sheep, and pig blood have the ability to control and affect metabolic, biochemical, and physiological processes within the human body and have the potential to be beneficial beyond just their nutritional values. Some properties of the bioactive peptides generated from slaughterhouse blood include antimicrobial, antioxidative, and angiotensin-converting enzyme inhibition. Blood can be divided into two main fractions. The first fraction is the plasma fraction, which is approximately 60 to 70% of the blood volume, and the second fraction is the cellular fraction, which is about 30 to 40% of the blood volume. The cellular fraction contains hemoglobin, which is responsible for over half of the protein content found in blood, and enzyme hydrolysis of the alpha and beta chains of hemoglobin produces several bioactive peptides. These bioactive peptides can be used within food and pharmaceutical industries. Large concentrations of these peptides inhibit a range of microbial growths and delays the meat rancidity process. These peptides could provide a natural alternative to synthetic ingredients added to food to delay the oxidation of lipids and prevent microbial growth that shortens the shelf life and threatens the integrity of the product. These peptides act as a natural alternative that could safely improve the transportation and the storage of food. Bioactive peptides provide the opportunity to be applied therapeutically to develop new prevention and treatment methods for chronic diseases. This can increase commercial returns for pharmaceutical companies, and bioactive peptides generated from slaughterhouse blood have ACE inhibitory properties that would serve as a natural alternative to synthetic drugs that often have negative side effects. Peptides with antioxidative properties could be used to combat free radicals and prevent diseases. The blood collection needs to be conducted using methods that will not contaminate or compromise the integrity of the blood. This can be done using a closed system collection method, using instruments such as a hollow knife, and a vacuum piping system to prevent the blood from coming in contact with surrounding air and becoming contaminated. Anticoagulants also need to be used to prevent clotting, and the blood needs to be kept at a temperature below 4 degrees Celsius until it is further processed. Crude neutrophil extraction is an effective method to produce an extract rich with bioactive peptides. An erythrocyte aggregating medium is used to isolate the white blood cells for removal from the blood, and various stages of centrifugation and filtration separate the blood constituents. The pilot scale level of production of bioactive peptides makes it difficult to purify the extract for individual peptides, but this allows for a synergistic microbial activity that is much more significant than the activity of an isolated peptide. The blood plasma removed during the first centrifugation can be hydrolyzed by plant and fungal proteases to produce its own bioactive peptides. After it is separated from the whole blood, it is centrifuged and the plasma is stored below -20 degrees Celsius. The freeze-dried plasma fraction is reconstituted using a phosphate buffer before hydrolysis occurs in a temperature-controlled incubator. Despite the potential for bioactive peptide generation, there are social concerns as well as safety concerns surrounding the use of slaughterhouse blood. There are health risks associated with collecting blood from slaughtered animals, such as an increased risk of transmitting diseases such as spongiform encephalopathies, more popularly known as mad cow disease. There's also concern around religious impositions and the inability of large demographics of people to consume blood-tainted food due to dietary restrictions, preferences, or allergies. In addition, blood products can produce strong flavors and aromas that could really disrupt the sensory experience of a consumer. The further processing of previously wasted blood would be beneficial to food, pharmaceutical, and meat processing industries. This would increase the value of the blood, it would generate a profit, and it would eliminate disposal costs, while also reducing impact on the environment and decreasing waste.